Okay, so our next talk is going to be given by Ercole, who is going to talk about practical steps for high-quality data, the DAQCORD tool.
Thank you, ladies and gentlemen. Thank you for the opportunity to come and speak. I should just say I'm speaking on behalf of a team of people involved in this enterprise, and actually a much larger cast of individuals and collaborators, though I'll present their names in detail at the end. So I've got the task at whatever time it is now to make data curation sound interesting early in the morning. I will try. I thought perhaps a good way to start doing this is perhaps to say something controversial. And there will be some controversy. And I think the most controversial thing I'm going to say here is that the CDEs, whilst absolutely crucial and necessary to ensure data quality, are not sufficient. Okay? It's a much more complex task than that. And of course, data quality underpins the whole enterprise of not just the science, but also data sharing and data reuse. So I'm a clinician. I'm also a data scientist. Data scientists are, of course, extremely cool. And in about 2016, I was working on the NINDS data set and thought I will, you can see my diary, on the 9th, I thought I'd come and have a look at the data. Nothing much happened after that. You'll see it's blank. Because we realized that, in fact, we maybe had a little bit of a problem. And one of the problems was that I told my friends that this is what I do. And in fact, what I was actually going to end up doing for 18 months was cleaning data. But actually, more seriously, this was a really complicated data set. Okay? There's sort of 2,000, in the center, there are 2,600 something variables. And there's lots of time points that these variables would happen. They were derived variables, all sorts of, it was actually really difficult. Biomarkers, genetics, wasn't really particularly dealing with the imaging, but really complex data set, okay? But okay, we can deal with that. And actually it should have been quite straightforward because this has been designed properly, okay? The InTBIR had used the common data elements, and there are lots of ways you can standardize your data. And if you don't like common data elements, there are other things you can do, and it's been designed properly. So this should be quite straightforward. But we rapidly realized that it wasn't. So let's think about some missing data. Missing data is important. We'd like to look at this as the data's coming in so that we can go and take appropriate remedial action when we find there are some problems from sites. This should be easy. Just find out where the data's missing. It's either there or it's not there. And sadly, it wasn't easy at all. And it's not easy because, in fact, the way that we recruit patients and follow them up over a period of time requires some skip logic. Not all data should be present at all times for all patients. Unless you codify that, which is not possible within CDEs, you can't know which patients should or should not have data elements, and therefore you can't even tell what's missing. So you can't take timely action to go and deal with that. Worse than that, as I start thinking about why some of this is actually missing, so we rapidly realized there were no patients with GOSE1. Well, that sounds like a problem, but of course these are patients who have died, and dead people don't come to follow-up clinics, and because of the way that sequentially we were recording data, this data never got put into the database. It's not that it's a problem, it just never got. Now we can solve this, we can post-populate in those patients who have died. We now need to know who has died, but of course death occurs in many, many different situations, many different data elements within this complex data set. So if you want to find out this is the structured query language, SQL query that you have to do within the database. It's not easy. Okay. And in fact, on that note, because this is complex data, it sits within a complex database, a relational database. So any time that you want to go and query that database or get some data out, you have to start writing some computer code. And I will come back to that at the end. We can start looking across sites. So I know what this is. This is calcium, but we can start seeing that, in fact, that calcium values vary across sites. And some people have used ionized, some have used not unionized calcium. And sometimes it all overlaps, like here. So we can't actually start to pull these things out. It's not that we haven't got a common data element. I know this is calcium, but we have a bigger problem within that common data element. Other things as well. This is some measurements within ICU. We have 2 hourly measurements of physiology and InTBIR in ICU, taken at different times, 2 o'clock in the morning, 4 o'clock in the morning, 6 o'clock, and so forth. But actually there's a discrepancy between what was intended and what was interpreted by those people putting in those times. Again, this is not a problem of knowing what those entities mean, but this is a problem of the annotation and description of those entities. Here is an extract straight from the CDE's website looking at place of occurrence of injury other, and we can see that it's typeset. We can have free entry text form. Let's look at some of that. This is some free entry from injury cause other within Center TBI. You can see there's some good stuff here. We've got head, hit, fall, fell, seizure, bite. There's definitely information, okay? But I don't know what I'm going to do with this information. How am I going to deal with this information? And here I've got a little extract of that particular data element, which I showed before. We've got 6-meter high audio device fell on, oops, I'll go back. 6-meter high audio device fell on patient over here. We've got fall from balcony, unhome, fall jumps, trees, ice hockey, llamas, bulls, cows, horses, motor, deer, skiing at martial arts. Here we've got plane crash, which is another problem because this is misspelt as well. So not only, okay, and then here I've got run over by cults. I really have no idea what that's all about. Another spelling mistake, I don't know. But the point I'm trying to make here is this is information here, but I have no idea what I'm going to do about it. And we can use magic words like natural language processing, but you're never going to fix this, okay? It's unnatural language processing, okay. And anyway, more seriously, the more that we started looking at it, the more we realized this is really complex, right? And there are also, it's not just the missing data, okay? And it's not that we don't understand what these data mean. The CDs do a great job of telling us what the data should mean, but it's the other bits about it that are tricky. So we start looking into variable inconsistencies and start looking at pairs of variables. So there are an awful lot of potential pairs of variables when you've got over 2,000 variables. Really, really difficult, okay? So that was my way of background. And really, I think from us and the similar efforts within TRAC, I think what we realized was, after sort of 18 months of this pain, was what we had seen cannot be unseen, okay? And now that I know how complex things are and how we know how complex things are, I will never, personally, trust any study that has not had an appropriate attention to detail to data quality. For these large studies, okay? I think we have. Okay, just to head that one off. But it's really important. It's not just about measuring the missing. And the question then is, how would one go about actually assessing the attention to detail? How would one go about teaching people to pay that attention to detail? Because we didn't really know this. Well, I mean, one strategy within the literature, within our reporting guidelines, and there are plenty of these with sort of Equator project. Do we have one really for observational? Well, we do. Strobe is probably the closest thing we have. But really, this is much more about sort of statistical methods and description of participants and data and so forth. Not really about how one goes about guaranteeing decent data quality, preferably how one goes about ensuring that prospectively, because I really would not like to have to do this retrospectively again, because it's never quite as good. So what were we aiming for? What emerged and what led to DAQCORD was this idea that we would try and provide several things. We would try and provide some criteria, some agreed criteria, against which one would be possible to demonstrate that your study design was likely to have good quality. A little bit maybe like Strobe, Prisma, Consult, so forth. So that was one aim. But the other aims were to actually produce a tool to assist people in thinking about how one prospectively would come up with a good study design. Okay? So partially, one would like this tool to potentially be able to demonstrate that a study has been designed well to fund potential prospective funders, but also to demonstrate that this was a credible study and that attention to detail had been put in. Because this has been a huge effort. I haven't listed the large team of people who were involved in data curation within Centre, similarly within TRAC. It's an enormous effort, okay? And any study that does not have attention to detail and curation, frankly, is not credible, I would argue. We also wanted to create a tool that was assist people in designing quality right from the start, and also, as a sort of result of that side effect, to improve data sharing through design documentation. Okay? And actually what we realized is that this, in fact, was domain generic. This is not necessarily anything to do with necessarily entirely with neurosciences. This could be spread to everything. And crucially, this is not a checklist. We're not aiming for another checklist. So DAQCORD was born, and this is what we did was use a modified Delphi approach to try and identify some key indicators. We involved stakeholders who I will list from large observational studies, and actually this is where neurosciences really comes in, because we've got some of the biggest studies that have really tackled this problem. We had 3 rounds. There was an online round, a face-to-face round that took place NIH 2018, and then a final round. It's registered with the Equator Network and read about it. So. And what we did, our methodologies, we assessed all of our concepts on 3 different domains. So whether or not the concept was valid, it was related to quality or not, whether it was feasible, in other words, you could actually measure or quantify it, and whether you could do anything about it potentially at the end of it. This sort of methodology has been used before, including within Center TBI. And we used a 5-point Likert scale. So by making this reasonably quantitative, it allows us to both assess the rating, but also the inter-rater agreement. And depending on whether something was rated as high or poor quality and whether there's good agreement, we could either take things forward or not between rounds. On top of that, we did do some free text, we took some comments, but we synthesized them thematically into examples, because what we realized, this is a very, very heterogeneous problem, okay? There's not gonna be one solution to any one concept. So what we want to do is provide actual examples. So here's a concept, here is an element, here's a parameter, something you should think about. Here are some examples of how one might, may not be appropriate to you, but how one might go about evidencing that you have addressed that parameter. So we did that. The nice thing about doing things numerically is you can write code and actually just automate some of the analyses. And Mike Jarrett's actually, and Vibeke Brink have been incredibly helpful in terms of actually producing a back end to actually to a platform for simulating this data. Started a literature review, generated about 106 potential items that we would start with. Went through our 3 rounds, and I won't bore you with the details, but we ended up with, at the end, with a final DAQCORD tool consisting of 46 items, which is a lot, and I'll come back to that point at the end, but the 46 final items. So I'm not going to go through in detail, you'll be pleased to know, but we're writing the paper up at the moment. And not all of these items, of course, will be relevant for every single study, okay? You pick and choose. Skip over that. So we'll be reporting in the next few months, you'll be able to read what these items look like, and we also have creating a living document which will sit on this website to provide hopefully guidance of good practice. What we would like to do is build and maintain an online tool, but that requires some build and it will require some maintenance in the future, so we'll have to think about how we might do that. So just to sort of finish off, 46 items, that's a lot. But again, let me stress, this is not a checklist. This is, people that know me know my hatred of checklists. I work on the air ambulance. My colleagues produced this mug for me. This is how much people know. This is not a checklist. What we try to do is produce something that is comprehensive and expansive, and that actually is guidance for people at the outset, rather than just some check. This is not an area that just lends itself to just ticking some boxes, okay? Not all of these elements will be relevant to all studies, so that doesn't matter. If it's not an imaging study, then the things we talked about imaging will not be relevant. That doesn't matter. And we cannot attempt to anticipate all the different solutions, and things will change in the future as well. So what we really wanted to do was encourage a sort of narrative approach to prospectively and looking at designing in data quality. So just to sort of finish, really, just to think of the wider picture, where does this fit in? We do have a problem, I'm going to argue. This was from 2006, and things like this sort of make me kind of laugh, but not in a good way. We all think reproducible science is important, okay? And I'm not arguing about that. Clearly, we think this is very, very important, okay? But this is only part, this is the tip of the iceberg. It really is the tip of the iceberg, okay? Because the data that we have, right, we may have good data, CDEs, these are crucial, but it's not versioned. Our assumptions aren't documented because there are a lot of assumptions that are going to taken from some raw data into something that we share with people, and our analyses are not robustly tested. If you don't believe that, might have seen this just in the last few weeks. This was a retraction in JAMA. I should say the authors have behaved impeccably here. What happened, this is a clinical trial, okay, randomized clinical trial. Well, they recoded in R, this is the code, right? They've got this alive, which is either a 0 or a 1, and you code it as a factor in your generalized linear model, okay, which recodes it into something internally, which is a 0 or 1, or in this case, a 1 or a 0. So that's unfortunate because now your alive dead has become dead alive, and you've not only got the wrong answer, but you've completely changed the causality or the association, the direction of the association. Now, this has happened because this is a computing, I mean, you know, we have operating systems, really complicated computer programs out there. There are ways in which you can avoid this sort of problem. This was, I was asked recently to look at some data, some positron emission tomography data, doesn't matter, just to do some, this is, I'm not expecting you to read this, but look, we've got here some reading data in stuff at the top, then we've got some data looking at the data, some data cleaning, retransformation, then some analysis. This is a computer program. Our analyses are becoming more and more like computer programs. So maybe we should learn some of the techniques and strategies that are used in the software development industry to actually document. Here's another one. This is some mimic data. I was doing this on the plane over. And I just wanted to get some calcium values out of mimic data sets. There's 200 and something calcium values here. I mean, nicely, we haven't got a problem with the different ionized and non-ionized. But here's some SQL just to get the calcium out, right, and then some lengths of stays. It's quite complicated. Did I do it right? How do I know I did that? How do you know I did that right? I'm going to publish somewhere. How do you know? Okay. It's not modular. So we should learn, I think, from software development And, we make a lot of choices and get in the answer, and that's why the DAQCORD tool actually encompasses all of these sorts of domains from development, testing, all the way through to reporting. We need to think about if we make these choices and these limit our reproducibility. So analysis is a process, a choice to how to remember, how to keep track, and how do we document this. And we should be thinking more about the software development tools and doing things in a much more structured way, which is, I think, where we hope DAQCORD will help. Just as an example, here was some outcome prediction, a paper we published. There's nothing particular special about this paper, but here is the GitHub repository. You can go in, you can go and read exactly how we prepared our data, and here you can even look at the Zenodo DOI to look at exactly everything we did. Okay? So this is a much, much bigger problem than just CDEs. Can we find other ways around this? And the very last thing, well, yes, there will always be some missing data, and there are smart imputation methods that we can use to deal with what's left over. Can we deal with the fact that there's dirty data? Well, you can build the clever models. This is a model that takes 13,000 completely unsupervised, completely unclean data and builds a good prediction model. But this sort of thing will never, ever, ever be a substitute for having decent data quality, particularly for the scientific domain. This works in the electronic health record. It's not going to work in the scientific domain. We have to have decent data quality built into our studies from the start. And if I've learned one thing from this whole data cleaning enterprise, is I never, ever, ever want to have to embark on a large-scale retrospective data cleaning and data curation process. This should be built in from the start, because it will save a lot of time in the future. So, in summary, this is hard, but it's really, really important. There are methodologies out there, but they will be unfamiliar to many of us within biomedical sciences, and we hope that the DAQCORD will be one of those methodologies that guides people to design in quality from the start. Thank you.